Hello students, in today's video we will talk about a very important central metabolic pathway, the Krebs cycle. It is a central metabolic pathway to yield energy. Let us proceed with the video. Tricarboxylic acid cycle or TCA cycle, or we call it as Krebs cycle or citric acid cycle. It is the second stage of cellular respiration, the three-stage process by which living cells break down organic fuel molecule in presence of oxygen to harvest the energy they need to grow and divide. This is the second stage. First stage is glycolysis, second is Krebs cycle, and third is ATC chain. We will study it in our upcoming videos. This metabolic process occurs in most plant, animal, fungi, and many bacteria. The site of Krebs cycle is a mitochondria in case of eukaryotic organism, but in prokaryote it takes place in the cytosol of the cell, because in prokaryote there is no mitochondria present in the cell. The TCA cycle plays a central role in breakdown or catabolism of organic fuel molecules, that is glucose and some other sugars, fatty acids, and some amino acids. So it is the central pathway where all these metabolites converge and produce energy in the resultant reaction in the Krebs cycle. Before this large molecule can enter the TCA cycle, they must be degraded, they must be catabolized into a 2-carbon compound called acetyl-CoA. So we can say that this is the central metabolic pathway. Once fed into a TCA cycle, acetyl-CoA is converted into carbon dioxide and energy. We will see it in detail in upcoming slides. In our food, we take carbohydrates, protein, fat, which get digested in the gut and absorbed by the cell as simple sugar like glucose in case of carbohydrate, protein, amino acid in case of proteins, and fatty acids and glycerol in case of fats. By the initial step of catabolism, they have to be converted into acetyl-CoA so that they can further enter into the Krebs cycle to yield energy. We will discuss about it in detail in our upcoming slides. It produced NADH, which is equivalent to which goes under ETC chain and produce ATP and release carbon dioxide. As we already studied that pyruvate, which is the product of glycolysis, has three alternate choices. It can enter alcoholic fermentation or lactic acid fermentation under anaerobic condition. What in case of when there is enough of oxygen, when there is a aerobic condition, this pyruvate is entered to acetyl-CoA, which get entered into the citric acid cycle and produce energy and carbon dioxide. Today's video we have to focus on the Krebs cycle. The person in the picture is a Hans Kreb, the person who has discovered this pathway. The very interesting fact about him is that in 1937, when he submitted his paper, they rejected it because they find it useless, but in upcoming year they find it very useful to understand the central metabolic pathway of living organism, and they acknowledge it in 1953. He got the Nobel Prize for this pathway. So let us proceed with the pathway. As we know that pyruvate is produced by the breakdown by the glycolysis. One molecules of glucose yields 2 pyruvate, and this pyruvate is 3-carbon compound which is oxidized to acetyl-CoA by the enzyme pyruvate dehydrogenase complex. This is a complex enzyme, and this conversion takes place in steps. The pyruvate, if you look at the structure of pyruvate, you will find that pyruvate is 3-carbon compound, and there is a acid group is Here, this acid group, which is COO, is released in the form of carbon dioxide, and one CoA is added to this, and as a result we get acetyl-CoA. In this reaction, one NAD positive is reduced to NADS2, which further go into the ETC cycle and ETC chain and produce ATP. We will talk about it in our upcoming videos. So what we get in this reaction, we get pyruvate. We convert pyruvate. We know that pyruvate is converted to acetyl-CoA. Acetyl-CoA is a feeder molecule for the Kreb cycle. This reaction is a link between glycolysis and Kreb cycle. So let us see the further step in Kreb cycle. In Kreb cycle, the oxaloacetate, which is 4-carbon compound, is condensed with acetyl-CoA to form citric acid citrate. This cycle is also known as citric acid cycle because the first molecule which is produced is citrate. That's why this cycle is also known as citrate. And if you look at the structure of citrate, it has 3 carboxylic group. That's why this is also known as tricarboxylic. 1 carboxylic, 2 carboxylic group, and 3 carboxylic. So this is also known as the tricarboxylic acid cycle, TCA cycle. This conversion is carried out by the enzyme citrate synthase, in which CoA is released and we get citrate at the end. And second step, citrate rearrangement reaction. First it produces acotinate, and acotinate rearranged to form isocitrate. The difference between the citrate and isocitrate is that it has OH group in second position. In this position, what in citrate it don't have OH in this position. So why this take place? Because the next enzymes which convert isocitrate into glutarate can convert isocitrate, but it cannot convert citrate. That's why it undergoes rearrangement reaction. This reaction, citrate to isocitrate, is conducted by the enzyme known as acotinase. The next step, isocitrate is converted to alpha ketoglutarate by the enzyme isocitrate dehydrogenase. Look at the structure of alpha ketoglutarate. Alpha ketoglutarate is 5-carbon. This is a 5-carbon compound. So what it means? It means it has to release 1 carbon. So carbon dioxide, this carboxylic group is released in the form of carbon dioxide. In this reaction, NAD positive is reduced to NADH positive, and NADS2, which goes into the ETC chain and produce energy, and 1 carbon dioxide molecule is released, and as a result we get alpha ketoglutarate, which is 5-carbon compound. This COO is added by the, this is a first acetyl group with carbon from the acetyl group. So we left with only 1 carbon from acetyl. So in next step, what happened? Alpha ketoglutarate is converted to succinyl-CoA. Alpha ketoglutarate is 5-carbon compound, but succinyl-CoA is a 4-carbon compound. So what happened? Here also this carboxylic group is released in the form of carbon dioxide, and resultant compound is found succinyl-CoA. This is the structure of succinyl-CoA. Here CoA is added, but the release of carbon dioxide take place. This reaction is catalyzed by alpha ketoglutarate dehydrogenase enzyme. In this reaction, CoA is added, while as NADH is converted to NADS2, and 1 carbon dioxide molecule is produced in this reaction. In further reaction, here 4 carbon is 2 carbon which is added by the acetyl-CoA is get oxidized to carbon dioxide. So we we are left with original 4 carbon atoms of oxaloacetate. So in the further reactions, succinyl has to be converted into oxaloacetate so that this cycle goes on. So what happens in next step? Succinyl-CoA is converted into succinate by the enzyme succinate thiokinase. This CoA is released and succinyl is in this reaction. One GDP is combined with inorganic phosphate and yield GTP, which is equivalent to the ATP, and one molecule and CoA is released. And next step, a small rearrangement takes place where succinate is converted into fumarate, because this, if you look at the structure, you will find that what is the difference between succinate and fumarate. Here is only one difference, that is addition of double bond between 2 and 3 carbon. And this bond is added because this 2 hydrogen molecule is added to the FADH, which is converted to FADH2. FAD molecule is converted to FADH2, and this reaction is catalyzed by succinate dehydrogenase enzyme. And next step, the fumarate is converted into malate. And what happened in this reaction, the double bond is converted to single bond, and addition of by the enzyme fumarase, and addition of water molecule take place, and in this position one OH is added. And further, malate is oxidized to oxaloacetate with the liberation of by the enzyme malate dehydrogenase. Here NAD is reduced to NADH2. So by this we have completed the Kreb cycle. This is the complete cycle which take place by one acetyl-CoA. We know that one glucose molecule produce 2 acetyl-CoA. So for the complete oxidation of glucose, this cycle should take 2 turns. So in this cycle we have to note that how many of NAD, FAD, GTP are produced. So these are very important to note. We will talk about the energetic of this Kreb cycle in next video. So for now you have to calculate the NAD produced. One NAD is produced when isocitrate is converted to alpha ketoglutarate. Second is produced when alpha ketoglutarate is converted to succinyl-CoA. And third is produced when malate is converted to oxaloacetate. So how many NAD we get? We get 3 NAD on one turn. And how many FAD we get? We get 1 FAD when succinate is converted to fumarate. So 1 FAD is produced, and 1 GTP is produced when succinyl-CoA is converted to succinate in this reaction. So we will get 1 GTP. This is inside the Kreb cycle. 1 NAD is produced, which is a linker in a linker reaction between glycolysis and Kreb cycle. So we take it in separately. This is the product of one turn, and we know that for the complete oxidation of glucose, Kreb cycle run in 2 turns. So we multiply 2 all this by 2. So at the end we get 6 NAD, 2 FAD and 2 GTP. So you have to remember this, how many ATP, GTP are produced. We will talk about energetic of Kreb cycle in next video. So the next is the significance of TCA cycle. In TCA cycle, complete oxidation of acetyl-CoA take place. As you know that acetyl-CoA is a 2 carbon compound, The two-carbon compound is completely oxidized into two molecules of carbon dioxide, so this complete oxidation of acetyl-CoA take place in a Krebs cycle. ATP is produced, you know that NADH, FADH, and GDP produced, which get converted into ATP in ETC chain and rearrangement. So, and ATP is the energy currency of cell. So the main function of TCA cycle is the production of ATP, the energy of the cell. The final common oxidative pathway where all the metabolic pathway converge at one point, as I already told you that integration of major metabolic pathway take place in this synthesis of new metabolites take place, and catabolism also take place. So it is an anaerotic and amphibolic pathway. Fat is burned on the wick of carbohydrate when there is a shortage of carbohydrate, glycogen. When you are in a starved condition, the fat is burned instead of burned, and it produces the energy required by the cell. Carbon skeleton of amino acid finally enter the TCA cycle when there is no fat, no glycogen, no carbohydrate. Then amino acids skeleton get into the TCA cycle and produce energy. This happens when extreme starvation, under the condition of extreme starvation. The citric acid cycle is amphibolic since in addition to oxidation, it is important in the provision of carbon skeleton for gluconeogenesis, acetyl-CoA for fatty acid synthesis and interconversion of amino acids. As I already told you that it is amphibolic, that is it takes part in catabolism and also in case of. With this we conclude our lecture. I hope you will find it helpful for your preparation. Thank you. Take care.